Hello everyone, and welcome to this new Gen webinar entitled Advances in CNS Drug Discovery, Focus on Neurodegenerative Diseases. This webinar was made possible through sponsorship from Wuxi AppTech. I'll be your host for today's event. I'm Jeff Bugaliscus, Technical Editor for Genetic Engineering and Biotechnology News, which is celebrating its 40th year, bringing you the latest information on advances within the life sciences industry. And in that time frame, we've covered quite a number of new drug discovery endeavors, many of which have led to therapeutics that have made huge impacts on patients' lives. However, for far too many of those years, pharma and biotech largely abandoned neuroscience research. Yet in recent years, thankfully, that dynamic has dramatically shifted as renewed interest, along with a surge in funding opportunities, have become available. Novel research projects and state-of-the-art technologies in genomics, transcriptomics, and proteomics have opened up promising alternatives for CNS drug discovery, the results of which are beginning to lead to the identification of novel, tractable, and translational targets for neurodegenerative diseases, something our group of expert panelists will discuss in great detail in just a few moments. But first, I want to introduce the moderator for our panel discussion today, Dr. Chris Rutan, who is the Director of Biology at Wuxi AppTech. Chris will introduce our other esteemed panelists for today's webinar, and then start the presentation by giving a brief overview of CNS drug discovery before handing things off to the rest of the panel. But first, briefly, I want to remind the audience to submit questions for our Q&A session that will begin right after the presentations. If you have a question for our panelists, you can send it in at any time during the presentations. You don't have to wait until the end. Just type your question into the Ask a Question box and hit submit. It's really that simple. Okay, with all that said, I'm going to hand things off. Chris, over to you.
Good day, everybody, and thank you for joining, and welcome to this webinar on recent advances in CNS drug discovery with a focus on neurodegenerative diseases. My name is Chris Rutan. I'm a neuroscientist by training, and I'm a director for biology at Wuxi AppTech. I will be moderating this session today, and over the course of 3 talks by experts from the field, we will explore examples of novel ways people are using these days to advance their CNS drug discovery programs. Today, we have Roland Burley, the VP of Drug Discovery at Cerevance, and he joins us for the first talk on target and hit finding and validation. Next, we'll have Jamie Bilsland, CSO of Astronautics, who will give the talk on the important role of astrocytes in neurodegenerative disorders. And finally, Deming Xu, who's an executive director and head of in vivo pharmacology at Wuxi AppTech, will speak about better animal models for Alzheimer's disease. But please let me kick off and give a short introduction into CNS drug discovery and the recent advances in this area. Neurodegenerative disorders are some of today's largest unmet medical needs. Currently, over 50 million people worldwide have some form of dementia, and numbers are expected to almost double every 20 years. Researchers still don't fully understand the underlying pathophysiology, and this makes diagnosis difficult, and clinical trials are long and complicated, making CNS disorders one of the most difficult fields to develop drugs for. As you can see in the figure on the top, Alzheimer's disease is by far the most prominent neurodegenerative disease, followed by Parkinson's disease, other dementias, and then by ALS and Huntington's disease. As mentioned before, little is known about the causes and the underlying mechanisms of the disease, and this is reflected by very poor attrition rates in this field of science. 99.6% of all clinical trials have failed to bring a drug to market, and over 90% of those are failing in phase 1 and phase 2 due to lack of efficacy and lack of safety. This has caused an exodus of big pharma and biotech out of the field of neurodegenerative diseases the last decade. However, on the right-hand graph, one can see the number of approvals of the last 5 years compared to the 5 years before per indication. And what you can clearly see is that the CNS field is actually the only field which has had more approvals in the last 5 years than in the 5 years before. So there might be some light at the end of the tunnel. So there have been some recent advances in CNS drug discovery. Recently, we've been leveraging novel state-of-the-art technologies in this field, and we're using more and more genomics, transcriptomics, and proteomics. We're applying new modalities in these fields, such as antibodies, peptides, proteins, or gene therapy. People are using human brain tissue at much higher rates, and we're using advanced iPSC stem cell technologies to do our research in. Furthermore, novel tractable biomarkers have been developed to guide our clinical trials. As a result of these novel technologies becoming more broadly accessible and available to the public, a renewed trust and interest from big pharma, biotech, and venture capital into CNS drug discovery has been observed. This can be seen in the graph on the right, showing the number of Series A deals and their amount of dollars involved. And what we can see is that after oncology and platform technologies, CNS disorders are the biggest field venture capital is investing in at the moment. Also in the bottom, you can see the pie chart where we see the number of chemical entities that had been filed in 2019. And it shows clearly that oncology is just as big as CNS and pain. So this means that an increased venture capital investment and big pharma deals have sparked interest into the CNS drug discovery. An example is Dementia Discovery Fund. This is a fund that funds several novel biotechs that are working on finding cures for dementia and neurodegenerative diseases. And two of today's speakers are from companies funded by the DDF, namely Cerevance and Astronautics. So until recently, the research into AD has strongly focused on the two proteins that are aberrant and overexpressed in the brain of patients, namely A-beta and tau. Over 30% of all clinical trials have focused on these two proteins. However, many of these preclinical studies relying on A-beta and tau rely also on faulty rodent behavioral models, and they have all failed to result into clinical success. Looking at the cartoon on the right, we can see that by using novel technologies, we've obtained a better disease understanding and better translation of the systemic, cellular, and molecular processes of aging preceding AD. This will help us identify new strategies and therapeutic targets, because we now realize that more and more factors and pathways, such as neuroinflammation, autophagy, vascular function, and oxidative stress, among many others, play a crucial role in the cause and development of dementia. And because we now have better ways to study and target these pathways, we have identified novel, tractable, and translatable targets for Alzheimer's disease drug discovery. So how can WuXi AppTec aid in your CNS drug discovery programs? Well, we offer a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industry worldwide to advance their programs and deliver treatments to patients. It's our mission to deliver comprehensive end-to-end solutions for creating, identifying, and supporting preclinical candidates from discovery to development with our integrated chemistry and biology research platforms. By means of an integrated CNS toolbox, we collaborate with our clients on projects in every stage of the drug discovery and development process. And we enable research across modalities, be it small molecules, antibodies, peptides, or gene therapies. Our CNS toolbox includes platforms for gene editing, omics, imaging, patient-derived iPSCs, as well as comprehensive in vitro and in vivo CNS disease models, including full DMPK and toxicology support. Across all these platforms, the process for identifying the most appropriate assays and models is enabled by sharing experience between our own seasoned drug hunters and the client's expertise. This will become clear also from the next presentations. In the upcoming 3 talks, we will give examples of alternative approaches to drug discovery in the CNS field, and more specifically in Alzheimer's disease. Roland Burley will focus on early drug discovery in Alzheimer's disease, focusing on target and hit finding and validation. Next, Jamie Bilsland will share his data from their hit to lead and lead optimization program on astrocytes as a novel target for CNS drug discovery. And finally, Deming Su will speak about the translational aspects of animal models and how to improve these. This has been an issue in CNS drug discovery for decades. So our first speaker is Roland Burley. He is a VP of Drug Discovery at Cerevance, a Cambridge, UK-based biotech, which is dedicated to the discovery of novel therapeutics for CNS diseases. Prior to joining Cerevance 22 years ago, Roland worked in drug discovery for over 20 years in biotech and large pharma in the US and in the UK. His specialty lays in medicinal chemistry. He did a PhD in chemistry at the ATH, and he was a postdoctoral fellow at Caltech. Roland co-authored over 100 peer-reviewed publications and patents. Hey, Roland, it's good to have you on the webinar, and I'm very much looking forward to your presentation and learning about the unique way that Cerevance approaches finding and validation new targets and hits for neurodegenerative disorders. Roland, please go ahead. The floor is yours.
Hi, Chris. Thanks a lot for your kind introduction. Next slide, please.
Yes.
Yeah, let me tell you very briefly something about Cerevance. So we are a biotech company based in Cambridge in the UK and in Boston in the US. We really are dedicated to the discovery and development of novel therapeutics for neurological disorders and neurodegeneration. And for this, we strongly believe that we need novel targets, and these targets are discovered by a unique target discovery platform, which we call NetSeq, and that's based on the human brain. The reason we use human brain and not animals as a starting point is probably best illustrated on the figure on the left side, where you can clearly see that the complexity of the human brain is far larger than, for example, a mouse or a rat here. Now, of course, after we discover the targets, the big challenge is to validate the target and to find chemical matter and progress towards molecules that are suitable for clinical trials. And we are building a robust preclinical pipeline with molecules that are well on the way to enter clinical trials soon. Next slide, please. Just very briefly about NetSeq, about our target identification platform. As I mentioned, it starts with human brain, with post-mortem tissue, and we have a brain anatomist here at Cerevance who dissects brain sections and identifies disease-relevant regions of the brain. We then take those and process those samples, and we isolate the nuclei, not the whole cells, because brain cells are quite difficult to sort. We then label them and sort them with FACS, the nuclei, and basically sort them into 50 to 60 different cell types, and then do a lot of deep transcriptomics. We look at about expression of about 12,000 genes. You can imagine after the data analysis, this leaves us with a humongous data set where we know which genes are expressed when, in which cell types, the relative expression of in healthy versus diseased. And so we get a massive data set from which we then select our targets. We go to the next slide, please. The target selection process is, of course, quite complex, and many things have to be taken into account. So we do look at changes of gene transcription in the disease. We look at specificity. Ideally, we want to select targets that are only expressed in relevant cell types and not everywhere to avoid unwanted side effects. Of course, novelty is a factor. A genetic link of the particular targets to the disease is highly desired. We of course use machine learning and all the typical tools to make these connections. We also look at tractability. Is a target tractable with a small molecule, or does need another approach, an alternative modality? Internally, we are dedicated to small molecules, but the platform in itself is agnostic of the modality, so other ways of interfering with with protein activity can be considered. And all in all this data together leads to a disease hypothesis, and then we need to, of course, test that hypothesis with therapies. Next slide, please. So as I mentioned before, the big challenge is, of course, we select the targets here. We need to validate this target. We need to understand which direction do we need to go. Do we need to up or down regulate? These are not trivial questions. And for this, we have several tools at hand. On the one hand, genetic manipulation like overexpression, knockdown, and CRISPR modifications. We use all of those tools to see whether and how we can affect the phenotype on a cellular level. But there is also pharmacology. If there are small molecular probes or tools available, then we, of course, synthesize them and use them. Otherwise, we have to go very aggressively and very early on into a hit discovery effort where we try and identify novel tools that can help us to understand this, the role of this particular target in the disease. And then that also, of course, leads us then to in vivo target validation, where typically we go in with small molecules into animal models. Next slide, please. So for the hit identification strategy, we have multiple ways of approaching this. Of course, we always try to pick the low-hanging fruits if that's possible. If there are literature tools, maybe they're even suboptimal, but they could serve as a starting point to make them better, more selective, more potent, and to understand on a cellular level at least what is the target, what's the target's role. One can do virtual screens. That works in some cases, particularly if a crystal structure is available, or we make homology models and build homology models and then go about it like that. And as another resource is of course a high throughput screen. And we've done quite a few different high throughput screens in the last few years. And that's quite often the most important starting point, because we do look for very novel targets, and often there is not much out there that can help us jumpstart the program. But one lesson that I've learned certainly is, if possible, we do like to explore multiple approaches in parallel. So maybe even if there is an early literature to run an HTS campaign quite early on to get to a better chemical matter that can then be improved further is a good thing. Once hits are available, or early molecules, orthogonal assays are really important for hit validation. And then what I'm a big believer in is to look for molecules in the right property space. So particularly because ultimately we do need CNS permeable molecules. So we have a big sort of aspect of this is then to look at all the typical characteristics that it takes for a molecule to get into the brain. And we do check for selectivity within family and off-target ADME profiling quite early on to understand the quality of our hit material. And then, of course, analog by catalog is something which is popular and helps us to do some rapid SAR generation. But more often than not, you have to start making molecules quite soon to optimize the compounds. Next slide, please. So this is the last slide. I just want to point out quickly how we then go forward from a hit to a candidate molecule, or first a tool and then a candidate molecule. So we don't do synthetic chemistry in-house. We have a large collaboration with WuXi and with other partners, where we outsource synthetic chemistry. We do scale-ups outsourced. We then do a lot of in vitro and in vivo study externally as well, starting from crystallography, in vitro pharmacology, DMPK, efficacy and safety. We do some in vitro pharmacology in-house, but also external. And WuXi is obviously a very important partner for all of those aspects. And then the data processing and then deciding what the next cycle is, then that we do mostly in-house. So this has gone quite well so far, and I'm happy to answer any questions. Thanks a lot for your attention.
Thanks, Ronald. That was really great. After this session, after the next two speakers, we will have a Q&A session in which we can discuss this presentation. Thank you very much. And in the interest of time, we move on to the next. Thanks, Ronald.
Thank you.
Our next speaker is Jamie Bilsland, and he's the Chief Scientific Officer of Astronautics, a biotechnology firm dedicated to unlocking the therapeutic potential of astrocytes in neurodegenerative disease. Jamie has worked in the field of neuroscience drug discovery for over 25 years, with a focus on neurodegenerative diseases and pain. He spent over 20 years in large pharmaceutical companies such as Merck and Pfizer, and he next moved into academic drug discovery as the head of the neurodegeneration biology at the Alzheimer Research UK-funded Drug Discovery Institute at UCL. He joined Astronautics in 2019. It's great to have you on this webinar, Jamie, and I'm looking very much forward to your talk. The floor is yours.
Thank you, Chris. So Astronautics is a small biotech company which formed about 2 years ago with a remit to understand and target astrocytes in neurodegenerative disease. Astrocytes are a supporting cell type in the CNS and are important for many aspects of CNS homeostasis. One of the key aspects of astrocytes is controlling CNS excitability at the synapse. If you move to the next slide, please. In Alzheimer's disease, there's a well-characterized neuronal hyperexcitability, which is observed early in the disease process and which is associated with loss of activity and disease progression. Now, there are several ways in which this hyperexcitability can manifest, either in a tonic increase in excitability or in phasic epileptiform activity. The tonic increase in excitability manifests itself really as increased activity in learning and memory tasks. This impairs network activity, as you can see in the cartoon on the lower left, and decreases connectivity across the CNS. Over time, the increased excitability early in the disease, predicts faster rates of clinical decline. Therefore, targeting hyperexcitability early in the disease progress will improve network activity and hopefully will decrease the rate of clinical decline. If you move to the next slide, please. So where do astrocytes fit into this? Astrocytes are also hyperactive in Alzheimer's disease models. So astrocytes signal by calcium waves, and in multiple Alzheimer's disease models, increased calcium waves are observed. What the increase in calcium activity does within an astrocyte is it modulates the support of the astrocytes for neuronal synaptic signaling, both by increasing the release of glial transmitters at the synapse, such as glutamate or ATP, and by decreasing the homeostatic control of glutamate and potassium and other ions at the synapse. So the net result of dysfunctional hyperactive astrocytes is dysfunctional hyperexcitable neurons. And this can be seen by modulating the activity of astrocytes in AD models, such as shown on the right here, wherein calcium hyperactivity in astrocytes in an APPPS1 model drives a neuronal hyperexcitability. If you move to the next slide. At Astronautics, we're interested in modulating this hyperexcitability phenotype, and we've identified a target which is selectively expressed in astrocytes and which modulates neuronal activity via astrocyte modulation. We've shown, as shown on the right, that the target is expressed in human AD brain, and you can see here it's expressed in the vicinity of plaques. And we can also recapitulate this expression in human iPS-derived astrocytes, and we can modulate the function of the astrocytes using small molecule inhibitors. We know the endogenous ligand for our target is secreted from activated microglia, So we carried out an HTS for modulators of this target in 2019, and we've worked with WuXi AppTec on developing our lead series since then, and we're currently at candidate nominations stage. This project's a collaboration between Astronautics and the Drug Discovery Institute, ARUK-funded Drug Discovery Institute at UCL, and WuXi AppTec have supported synthetic chemistry, primary pharmacology, and all of the ADME and PK that we've done on this project. If you move to the next slide. In terms of the efficacy of modulators of our target, we've generated some nice data within UCL, which demonstrate that activation of our target using a selective agonist drives excitability in transgenic AD mouse brain. And you can see here on the top panel here, the effects of an agonist and an antagonist on action potential firing in an APP-NLF knock-in mouse model. In contrast to the wild type, where addition of the agonist does not drive action potential generation. On the bottom, we can see that when we add an agonist to our target, we can see an increase in excitatory signaling, and we can block the endogenous increase in hyperexcitability with an antagonist to our target. So overall, if we take these data together, they provide strong evidence that our target, our astrocyte target, is expressed in neuronal hyperexcitability in Alzheimer's mouse brains, that if we activate it, we see an increase in excitability. If we inhibit it, we see a decrease in excitability. If you move to the next slide. What we're interested in primarily in this project at the moment is translation. So what we've put together is a translational plan where we're trying to identify tissue and physiological biomarkers for translation. I'm going to say a little about our efforts in physiological biomarkers. What we ultimately want to do is define the exposure to target pharmacology to physiology relationship preclinically with a view to identifying a physiological clinical biomarker. And we've done some EEG pilot studies to assess neuronal activity changes, and we're currently following up these experiments in AD transgenic animals. To do this, we've been using a tool compound. You can see the properties of the tool compound on the right here. I'm not going to walk through them, but it's a potent, orally bioavailable and CNS penetrant molecule with a decent half-life. Go to the next slide, please. So I'm going to show some EEG data that we've generated in naive rats. What we can see is on oral dosing of our compound, we see a significant but small effect on wakefulness with no effect on locomotor activity, but a significant reproducible dose-dependent increase on delta power and a significant reproducible increase in wake gamma power. And I've highlighted the delta power changes in non-REM in the data on the right here. So what you can see is two experiments, one a single dose study, the second 3 doses. What I'm highlighting here is the efficacy of the compound over the first 3-hour time bin post-dosing. With the compound, we see a physiological change out to 9 hours post-dosing. But what you can see is a significant, reproducible, dose-dependent effect on CNS function. We're currently carrying out some EEG studies in an AD transgenic mouse. Our baselining studies have shown detectable EEG changes in an APP-PS1 mouse line, which have previously been shown in the literature to have hippocampal hyperexcitability. And we'll view this as a proof of pharmacology study in a pathological setting. If you move to the next slide. So just to summarize, astrocytes really have been a neglected cell type in CNS drug discovery in spite of their critical role in CNS homeostasis and the fact that they're known to be dysregulated in neurodegeneration. We've identified a target which is expressed on astrocytes and shown that this target is involved in neuronal hyperexcitability. And we've shown a role for this target in Alzheimer's disease models. We've shown that antagonism of our target improves synaptic function in an AD mouse line. And we've also shown that CNS penetrant antagonist of our target causes changes in EEG, a potentially translatable biomarker. And we're currently doing work to explore tissue and soluble biomarkers and EEG changes in AD transgenic mice. We're at the point of candidate nomination in this project now, and we're planning to raise a Series A later in 2021 to support clinical proof of mechanism. If anybody wants any further discussion on this, I look forward to any questions in the discussion sessions, or you can email me directly at the email attached below. Thank you for your attention.
Thanks, Jamie. That was excellent. I'm sure we'll have some time afterwards for questions and discussion. We now need to move on to our last speaker of today. His name is Deming Su, and he's an executive director and head of the Department of In Vivo Pharmacology at WuXi AppTec. Prior to joining WuXi in 2010, he has 15 years of R&D experience in the pharmaceutical and biotech industries and academia. His current research interests include in vivo pharmacology of NASH and hepatic fibrosis, infectious diseases, respiratory and neurodegenerative diseases, and aging. The WuXi teams under his management have contributed to R&D and preclinical programs that have advanced over 20 compounds to preclinical candidate and regulatory filings, several of which are now currently in clinical trials. It's great to have you on the webinar, Deming, and I'm looking forward to hearing more about the advances in animal models and readouts for AD. The floor is yours.
Thank you, Chris, for the generous introduction and for organizing this webinar. I also want to thank Roland and Jamie for their presentation and for laying the ground for my presentation. I want to give a brief account of what my team has been focusing on, applying electrophysiology, EEG, to animal model of Alzheimer's disease. The three primary pathological lesions associated with Alzheimer's disease are the accumulation of extracellular A-beta plaques, activation of microglia and astrocytes, and eventually the accumulation of intracellular phosphorylated tau protein tangles. How these pathological lesions lead to the clinical manifestation of Alzheimer's disease is not well understood. The deterioration of brain activity in the progression of Alzheimer's disease is the missing functional bridging between molecular pathology and the clinical outcome of this disease. So conversely, if such a functional bridging exists, it could be used to not only monitor the progression of disease, but also to monitor the efficacy study of test compounds. So my group did a literature research and found this indeed is the case. The brain activity of Alzheimer's patients are compromised. In the healthy individual, the EEG oscillation of various frequency are highly coordinated. In the case of theta and gamma oscillations, their respective phase and amplitude are coupled in the healthy individual, known as PAC. And this PAC is compromised in patients of mild cognitive impairment. And this was further compromised and completely disappeared, completely gone in patients with advanced dementia. In another paper, earlier paper, it's shown that hippocampal task-related activities are compromised in patients of advanced dementia. So we want to duplicate these clinical findings in an animal model of Alzheimer's disease, the 5X-FAD mouse, which is a transgenic model containing 5 Alzheimer-related mutations in 2 different genes. And shown in this slide is a summary of the time course of various pathological findings at different stages, different time points, from 3 months all the way to 14 months. And I'll come back to this slide a little later, okay? We then also did analysis of the A-beta 40 and A-beta 42 at 10 months, and also did the Morris-Watermaze analysis test at the 6 month and 10 month. So the results are largely consistent with what is reported in the literature. In particular, we found that the model animal are significantly compromised in cognitive behavior in the Watermaze test at the age of 10 months, not before. So we then look at the hippocampal local field potential in this model animal. So shown on the left is the power spectral density, the raw data, and we can see significant changes in the PSD as early as 6 months. And in particular, theta and gamma oscillations are most susceptible to these changes and as the animal get older. So when we look at the PAC as depicted here by the heat map in this slide, okay, and it is rarely observed in wild-type animals. However, the loss of PAC is evident as early as 4 months in the animal model, and it got deteriorated with time and almost completely gone at the age of 10 months old. And changes in the PAC modulation indexes are statistically significant at 4 months of age. So we put all these data together and come up with this atlas of the 5X FAD animal model. So the accumulation of A-beta plaque was seen as early at the earliest detection point, in this case 3 or 4 months. Okay, and microgliosis is also happens around the same time. The accumulation of phosphorylated tau protein tangio start a little later. There's a delay. And in terms of electrophysiology, EEG, okay, the loss of PAC was seen as early as 4 months. And likewise, the change of local field potential, the PSD part, okay, is also seen around 4 months time. However, the cognitive impairment only became significant around 10 months of age. So there's a window of 6 months between the EEG change seen in this animal and the significant cognitive impairment. And we want to explore this window to testing efficacy. So I want to show a case study of how the EEG predicts efficacy. The compound we used is FPSZM1, which is an inhibitor of the receptor for the advanced glycation end product known as RAGE. And it is thought to be a receptor of pattern recognition. It's implicated in various inflammation processes, including the neuroinflammation associated with A-beta amyloid in the Alzheimer's patients. So when we first dosed the animal for 1 month, and then we looked at the EEG result, we saw that there's a significant improvement in the PSD at 1 month, after 1 month's dose, okay? And this is shown on the left side of this slide. And when we looked at the PAC, we saw that in the model animal, there's significant impairment, loss of the PAC. And the treatment of this compound for 1 month was able to reverse marginally, though, this coupling, particularly at the high frequency, gamma high frequency, okay? So if you look at this, if we break down this coupling into by two segments, and the low frequency and high frequency according to gamma oscillation, we see that in the high frequency oscillation, the PAC modulation index are significant in this case after 1 month's treatment. So we speculate that with 2 more months treatment of this compound, we should be able to see efficacy. So in the follow-up study, we did 3 months, actually it was first month and then plus 2 more months treatment. So that's total 3 months of the FPS treatment. And after that, we did the water maze test. And it's clear that the treatment yield a significant efficacy in this cognitive behavior. So we were very pleased to see this data. And in summary, what my group did is we discovered that the EEG is a mean, perhaps a reflective of the brain function. And if we use LPF or and TG-PAC, it could be used as a functional bridge between pathology and behavior. And we also demonstrate that there's a window between the 2 aspects, the starting and the endpoints, and the EEG results could be predictive, and when used the efficacy test. So I think I will stop here and hand the floor to Chris. Thank you.
Thank you, Demeng, for this exciting presentation. Let's hope these new readouts will help better translate preclinical findings into clinical trials. In the upcoming Q&A session, we'll discuss your talk as well as the other two talks from Roland and Jamie. The Q&A session will start in a couple of moments. For further information, please visit our website, contact one of our business developers, or connect with us over LinkedIn.
Gentlemen, excellent presentations and insights into drug discovery for neurodegenerative diseases. We really appreciate your time, and thanks for sharing all that information with us. To the audience, our Q&A is coming up in just a moment. So if you have a question for our panelists, now's the time to send them on in. It's really simple. All you need to do is type your question into the Ask a Question box and then hit submit. Okay, it looks like we have a bunch of questions already, so that's great. If you have a question, don't hesitate, send it on in. Bear with us for just a moment now as we make the transition into the Q&A session. All right, everyone. Thanks for joining us for the Q&A session. Really appreciate it. We got a bunch of questions for our panel here. Just want to make one note beforehand. We saw, some of you may have seen that Roland's title was a little bit different. We did have an error there that we've rectified. He is a VP for drug discovery, not the CSO or CEO if it has been that way in the past. So just wanted to make that note. And we have a bunch of questions. So let's turn it over to Chris for the first question. Chris, one of our audience members would like to know, they say you talked about supporting integrated CNS drug discovery programs, but is it also possible to do just chemistry or biology assays?
Hi, Jeff. Yeah, thanks. Of course, the simple answer would, of course, be yes. The way we're built up at WuXi AppTec is that we have two departments. We have WuXi Chemistry, where you can do, which is a world-leading chemistry service. And we can do basically anything you require for chemistry, including the synthesis of small molecules, oligonucleotides, or peptides. And on the other hand, we have a business unit of WuXi Biology, where we support our clients in the discovery and development of all your therapeutics. And here we have expertise in all different kinds of indications, in oncology and immunology, infectious disease, cardiovascular and metabolic, but also neuroscience. And then regarding this CNS platform, you can choose whichever service you need. So you can build a full integrated service, including chemistry, including SAR, including hit finding, or you can just select a specific assay which you would need and then pay for it in a pay-for-service kind of way. So in the CNS platform, we have a broad portfolio of in vitro and in vivo assays, and you can go all the way from simple screening assays, binding assays, functional assays, all the way into microglia and astrocyte biology, oxidative stress assays, A-beta aggregation assays, and also into the in vivo models where we have a broad portfolio of all the different mouse models, the corresponding EG and EFFICACY readouts, and also the histopathology and biochemical readouts you may need to assess your compounds. So it's basically any way you want to do it, either if it's just a fee-for-service and one specific scientific question you have, we can support you with that, but we can also go for a full-fledged program, including experienced chemistry or biology project leaders and everything in between. So that's basically what we would like to offer.
All right, thank you, Chris. Roland, we have a question for you from Katarina, who starts off by saying, thank you very much for your interesting presentation. And she says, regarding the NET-seq methodology, brain tissue needs to be fresh, correct? This is not easy to get, even when having collaborations with hospitals. We usually don't know how fresh and in what condition samples are. How do you envision to sort of counteract this challenge?
Yeah, that's a really important question and points at the heart of our technology. So early on, when NET-seq was developed, there were actually several innovations, and one of which was that we are harvesting nuclei, and we only look at transcripts in nuclei. And what we found is that the nuclei are much better preserved, and the transcriptome is much more stable in that sense than in the whole cell. I should also say that early on the team has performed sort of
studies with rodents with various post-mortem intervals, and extending this to from minutes to several hours, and I found that the samples are actually very stable. There's very little changes apart from some as you might expect, some sort of acute heat shock proteins or things like this that are very sensitive to stress. Most of the transcripts are actually very stable, and even when you freeze the samples and then get samples from brain banks afterwards, this is quite robust. So a lot of controls have been performed, and we're quite confident that what we're seeing is real. Thank you.
All right, thank you, Roland. Jamie, we have a question from you, from Anne, who says, high efflux and high LOD or high ppb for tool molecule would suggest limited uptake into the brain. Is that the case in vivo?
Thanks, Anne. In short, that doesn't appear to be a problem for this tool molecule. So we have done in vivo PK studies in both mouse and rat, and in both cases for this molecule, the brain to plasma ratio is about 0.8. So we get about 80% in the brain compared to the plasma. You're right to point out that we have a high log D, and in fact we've had a high log D in this series throughout. We believe that the high lipophilicity in this series may in fact be helping with the brain to plasma ratio, because we think the compounds may be preferring the lipophilic environments of the brain, because some compounds within this series we actually have brain to plasma ratios total and unbound, which are greater than 2 or 3. One element of your question I'm not sure about is the high efflux. So the efflux that we saw with this tool molecule was 3.5. Typically, we think an efflux ratio of less than 5 is likely to be CNS penetrant. Less than 2 is highly likely to be CNS penetrant. So we don't believe that we have much PGP efflux with this compound, or indeed this series. One thing that the high lipophilicity has brought us, though, is high non-specific binding, which caused a few problems with our screen sequence, but also high plasma protein and brain tissue binding, which is greater than 99%, both for this tool molecule and for later compounds from the series.
All right, thank you for that, Jamie. Deming, we have a question for you. Yang Ling would like to know if you can comment about EEG as a potential biomarker for AD in the clinic.
Thank you very much, Jeff. Yeah, this is indeed a very interesting question. So the study we performed in the animal model was based on what was observed in the patient. Okay. However, the way the flow of the event is that patient would first diagnosed with Alzheimer's disease and then use the EEG to confirm it, right? To make the observation. So really, the question regarding the potential use of EEG as a biomarker is actually you have to do the proof of concept. Coupling associating the progression of the Alzheimer's disease with the EEG signal or the lack of, and as well the improvement of the drug treatment. And if you can link these two aspects at least, okay, plus a little bit of mechanistic study, and probably you should be able to use this as a biomarker for both monitoring the disease progression and also as a way to monitor the efficacy study. Okay, so what we're doing right now is actually we're doing a very comprehensive study using different drugs and different models, try to expand this observation, beyond the initial experiment with 1 or 2 drugs. Okay, so and as well, I would like to expand this to the human primates and see how as the animal gets old or develop dementia, okay, how this EEG changes. And likewise, we wanted to see association. And of course, once it gets to clinic, it's getting complicated. So you need clinical studies to support this, okay? Thank you.
Can I come in? Can I come in on this question as well, because it's a very interesting question. I mean, there have been some studies published recently, and some ongoing trials which are using EEG as a biomarker. An example might be the low-dose levetiracetam trials in EEG. But what I thought might be interesting to raise in this forum is that this is an area which the Dementia Discovery Fund is interested in, and which Alzheimer's Research UK funding charity is interested in. The Dementia Discovery Fund is working together with some other partners and have put together a really interesting company called Cumulus Neuro, which is developing a home use EEG headset together with a tablet-based device, which can take EEG measurements from patients and couple the EEG readouts to the response of patients to gamified cognitive tests. And this is rolling out now, so there are early trials going on now with a view to it being used in phase 1, phase 2, and possibly even large-scale phase 3 trials in the future. And it's a very interesting technology to watch.
All right.
Totally agree.
All right. Thank you. Roland, we have another question for you. From Fred, who wants to know or starts off by saying, starting from human sample, you must strongly believe in human-specific aspects of disease pathogenesis, and your targets might well be human-specific. Thus, do you still use mouse models for target validation, or do you completely get rid of this aspect in your pipeline?
Yeah, that's a really good question. First answer is yes, we absolutely believe that there's something specific in the human disease pathogenesis. Neurodegeneration, this often takes many years until the disease manifests. It's very difficult to mimic in any disease model, preclinical disease model. I'm sure you're aware of that. Unfortunately, we do still need some sort of preclinical target validation or maybe, I mean, my personal view is actually target engagement. Preclinical target engagement is really important to show. So if you have like some sort of way to show that the compound ultimately engages in the target and the pathway, and then clearly it's also safe, because you want to avoid that there's any unexpected on-target toxicity that is not tolerable in humans. But once you have a safe compound and we know it engages the target, then ultimately the efficacy will be shown in the clinic. But if we do aim for certainly a preclinical efficacy readout as well. So it is still important, yes.
All right. Thank you very much, Roland. Jamie, another question for you, a pretty specific one and a good one. And I always would like to know, why have astrocytes been overlooked to date in CNS drug discovery?
Thanks. So that's a curious question. So as those who follow the field will know, there's a switch from a neuron-centric view to a more neuroinflammation-based view in AD, which has been largely driven by the cluster of microglial targets which have come out of GWAS studies. But the astrocytes have been largely overlooked. And I suspect this is partly because there's a prevailing view that astrocytes are glia, they're glue, they're relatively inert bystanders within the CNS, but that's simply not the case. So astrocytes are really fundamental cells for supporting CNS homeostasis, whether it be provision of metabolic support for neurons, or supporting synaptic transmission, or supporting the blood-brain barrier. We feel there's a real source of untapped targets there, and the field has, by overlooking these cells, largely missed a trick. But I guess the proof will come with developing astrocyte-based targets and taking them to the clinic. But for a cell type that's so fundamental to the homeostasis of the CNS, it seems a curious omission.
All right, thank you.
May I chime in as well?
Sure, absolutely.
Yeah. So yeah, this is an important aspect. The neuroinflammation is an important aspect in the progression of Alzheimer's disease. Okay, what I would call the damage-associated molecular pattern associated with Alzheimer's disease is largely unknown. So the process, Roland mentioned, Jamie mentioned, is actually the reason I think it's being neglected is that we really don't understand. We don't have much understanding of the clear pathway associated how this damage-associated molecular pattern triggers the inflammation. Okay, so that's why I think that's at least one of the reasons that contributes to the neglection of this important aspect of Alzheimer's disease, the astrocytes. Thank you.
All right, thank you. And stick with me there, Deming. We have a question for you, actually. Hafiz would like to know, does WuXi do NHP studies as well? And if so, are they agnostic of drug modality?
That's very good. We do have two actually sites, animal facilities dedicated to non-human primates. Okay, and the total holding capacity is over 1,000 non-human primates of different ages, and we actually have a collection of aged monkeys. Okay, so of different stages, and we're looking for natural occurring diseases. It is disease agnostic in the way that we're looking for natural occurring diseases. Okay, right. So we can we find metabolic diseases which can be induced, cardiovascular, and we also see monkeys with Parkinson's diseases, for example. Okay, Alzheimer's is a hard one. Okay, I have not seen any at least in our collection. Okay, but dementia is something we're interested, and we're actually developing a program trying to monitor the phenomena of dementia, okay, in human primates. Not doing behavior tests, okay, but rather than observing the behavior of the animals. So this is something what I call this automated AI-based behavior studies. So yeah, it is disease agnostic in the way that we're looking at. So we cover pretty much all kind of diseases, natural occurring disease, all kinds. Okay, and we're building up the capability of age-related disease, and neurodegenerative definitely is one of the priorities. I will stop here. Thank you. Yeah.
All right, thank you, Deming. Next question for Roland. Roland, Anne would like to know, do you use your platforms to repurpose old marketed or failed drug compounds?
We haven't used it for that specifically, but it's a good approach, and we certainly have talked about it. Why have we not done it? It's frankly lack of capacity. I think it's a very interesting thing to do, and it makes sense. But clearly, to make our stand, we do prefer to work on something novel and really have a fresh approach. But this is absolutely something we will likely consider in the future.
All right, great. Jamie, question for you. Iva from Auburn University says, thanks, wonderful presentation to all of you. It's a little bit of a long question, Jamie, so bear with me for a second. She says, would you consider glial hyperexcitability to be the major pathological target in terms of novel drug discovery and development? Also, what are some cross-links with neuroinflammation that could possibly work as biomarkers for both? Thank you.
So taking the first question first, so glial hyperexcitability as the major pathological target in terms of novel drug discovery. I think it's a major pathological target. Clearly you see neuronal and glial hyperexcitability early in disease. There's some evidence that that may be, certainly there's evidence that where you see the hyperexcitability in the clinic, you see a faster disease progression. It seems likely that interfering with that, therapeutically altering the progression of the hyperexcitability will have a clinical benefit. Now, whether it's the major pathological target is a separate question, because clearly what you have with AD and other neurodegenerative diseases are multifactorial diseases, and you know, I think that there are several strands that we could take, and even if you're focused, as we are, on a specific cell type, so even if you're focused on astrocytes, then there are other pathological targets that one could take. In the rest of our portfolio, for example, we're interested in modifying the provision of metabolic support for neurons by astrocytes in one of our other projects. Neuroinflammation, as you mentioned in the second part of your question, is also, I think, a huge potential source of therapeutic targets, both for microglia and for astrocytes, which are inflammatory cells in their own right. So I think that there are quite an array of targets, all of which are potentially valid. The hyperexcitability target, I think, is attractive because it's early in the disease process and has potentially symptomatic and disease-modifying benefit. In terms of the cross-links with neuroinflammation, I mean, in terms of biomarkers there, I mean, we're interested in looking at some of the astrocyte biomarkers, GFAP, YKL-40, soluble biomarkers. Actually, we're in the middle of a study right now with our lead project, where we've carried out a long-term chronic dosing study in the therapeutic range, and we're working with Olink and with Henrik Zetterberg at Gothenburg to look to see what biomarkers are changed by our lead molecule for our lead project, and it'll be interesting to see how this work pans out. We don't have the data back from that yet.
All right, thank you for that, Jamie. Deming, we have a question for you from Sung at LSU, who'd like to know, is it difficult to show extracellular A-beta with 5x FAD mice? And how do you use extracellular A-beta with 5x FAD mice?
Okay, yeah, thank you, Jeff. Yeah, so we actually looked at the histopathology-wise. Okay, you can look at at higher resolution. Okay, you probably can look at where the signals are. But more importantly, I think, you know, we can look at in the animal model, the plasma level of A-beta, and also the CSF level. Okay, so these are good indication of extracellular extracellular levels of A-beta. Okay, so I think it's quite significant. We actually have a time course of these extracellular level, I mean the CSF and the plasma level of A-beta, and they're highly 40 and 42. Okay, they're highly consistent, aligned with the pathology and the behavior. So, thank you. Yeah.
All right, thank you, guys, for that, Deming. Roland, we have a question for you. Igor would like to know, in addition to small molecules, are protein biologics, like antibodies and nanobodies, etc., also used in your target validation workflow? And if so, how?
So there is certainly a place for, particularly for antibodies. What we do is try to see whether there is a co-localization of the target and the cell in which it is expressed with for example, with pathology like plaque, for example, in Alzheimer's, if you will, or Lewy bodies or so. So if you see those co-localizations using antibodies, that's definitely good. In terms of modulating the target, which is maybe what you are referring to, we are a small molecule company, so when we make tools, they will be small molecules. The NetSeq platform itself is modality agnostic. In principle, one could go about this with antibodies, ASOs, or anything. And in our workflow, when we can, we use molecular tools, but we also do overexpression or knockouts or CRISPR, sort of catalytically inactive forms of the protein or so. But that's about the tools that we have to handle. I hope this answers the question.
All right, thank you, Roland. Next question is for Jamie. Jamie, Hi would like to know, does targeting your target in astrocytes affect neuroinflammation in your model?
Thanks for the question, Hi. That's a great question, and it's one that we're actively looking at the moment. We believe that our target will have effects on the astrocyte modulation of synaptic transmission, but we believe that it may also have an effect on the astrocyte component of neuroinflammation. So as I mentioned earlier, and as I'm sure you know, astrocytes are themselves inflammatory cells within the CNS, and certainly there's a literature from dating back a number of years on microglial-induced astrocyte reactivity. We don't necessarily believe that the target will have an effect on the microglia, but we believe it might have an effect on astrocyte reactivity. So we've got some in vitro data that we generated where we took a tool compound which interacted with our target and then stimulated astrocytes in a co-culture model, and we could see small decreases in complement C3 and LCN2 and STEAP4 and classical markers of astrocyte activation. What we're doing at the moment, as I mentioned earlier, we're running a large chronic study and in an AD model, an APPPS1 model, and in that we're looking at microglial inflammation. We're not necessarily expecting to see a change there, and we're looking at markers of astrocyte reactivity together with CSF and plasma biomarkers. And we're throwing quite a wide net with Olink, looking at a range of cytokines and chemokines, and along with more traditional inflammatory markers. So that'll be an interesting data set when it lands. To answer your question directly, we don't necessarily expect to see an effect on microglia. We do think there's a good chance that we'll see an effect on astrocyte reactivity based on our in vitro data, but we're actively exploring that at the moment with a large chronic dosed in vivo study.
All right, thank you, Jamie. And it looks like we have time for one last question for Roland. Roland, question from Younghwa who'd like to know, with regard to your NetSeq platform, is there a method you use to tease apart the expression pattern that is the outcome of the disease from the expression that is actually a driver of the disease?
Yeah, that's a hard one to answer because, I mean, NetSeq itself, what does it teach us? It teaches how specifically a target is expressed, which cell type it is expressed. It teaches us whether it's up or down regulated in the disease state. Of course, mostly towards the end of the disease, because most of not all patients die sort of from the disease, but a lot of them do. So we mainly get sort of the late stage part there. It's clearly not possible just from that data to say whether a target is a driver or of the disease or not quite the cause. And we do emphasize a lot on formulating a mechanistic or a hypothesis that we can then follow in terms of the pathway, not just like is that pathway likely to be involved very early on in the etiology of the disease. Is the genetic link, of course, that's all of those factors are taken into account when we consider whether we really hit something fundamental, something central to disease. But I would like to also point out, we talked about neuroinflammation. My personal belief is that neuroinflammation is not necessarily always disease causing, but it kind of spirals the whole brain or the brain cells into an exaggerated status, and microglia get hyperactivated and so on. And if you can attenuate that kind of dam state, will you then, even though this may not be disease causing, it certainly contributes to the progression. Can you then slow down disease progression? So I think, at least my understanding is that these are highly unanswered questions. it's not. I wouldn't claim that NETseq points to the driver or the cause of the disease per se, but it certainly unravels pathways that are involved. But it takes more than just that to understand really where do we go with our and which targets do we engage with. I hope this answers the question a little bit. It is a difficult question to answer, of course.
All right, thank you, Roland. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the GEN website at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or as we always recommend, you can feel free to forward the link to any of your friends and colleagues who you think might be interested. I'd like to thank Chris, Roland, Jamie, and Deming for their amazing presentations and very great in-depth discussion here. And I'd like to thank you, the audience, for your attention and thoughtful questions. Very special thanks to WuXi AppTec for sponsoring this webinar. Hopefully, we'll see you again at another GEN webinar in the near future. Goodbye for now. Everyone stay safe and healthy and get your vaccines.